Hello, everybody. My name is Joseph Giancaspro. I am a senior biochemistry major here at Iona's honors program. I have been a member of Project Symphony, which is a research group ran by Dr. Lee, for the past 4 years. Today, I'm going to be talking about one of the many projects I've been a part of, and that is how caffeine has an effect on a model cell membrane. So why did we want to do this? As you may know, as most of you do know, caffeine is a commonly used stimulant drug in American culture. And we usually ingest this via the consumption of coffee. On average, an American consumes 3.1 cups of coffee a day. And we do this because it gives us a sense of wakefulness. But what you may not know is that many drug pharmaceutical companies incorporate caffeine into their drug formulation as a drug adjuvant. So they use this to increase the efficiency or efficacy of these drugs. And three examples are Excedrin, Midol, and Anacin. So our goal was to understand how caffeine interacts with our cell membranes. So we know that as a normal individual, that we take caffeine and we get a sense of wakefulness, but we usually don't have a full understanding of how caffeine interacts with our biology. There has been published literature and findings that suggest that caffeine can influence the physical properties of our cell membranes. But in specific, our research looked at how caffeine can affect the thickness of our cell membranes. And this is of importance because the thickness of our membranes can alter the activity of membrane proteins. And if we have altered membrane proteins, it can lead to diseased phenotypes. So by understanding if caffeine can affect membrane thickness, we can understand if it has an effect on membrane protein function. So in order to do this, we had to do two things. Number one, we had to mimic a cell membrane. And to do this, we used the droplet interface bilayer method. So to do this, we submerged two aqueous droplets in an oil containing lipids. The lipids oriented themselves around the water droplets. And when we brought these two water droplets together, we got a simplistic version of a cell membrane, as you see by these red boxes. Once we have this system, we needed to design a device where we can incorporate the system and measure the bilayer or the membrane's thickness. In our system, instead of directly measuring thickness, we measure the capacitance of the membrane. I'm not going to go into the physics of capacitance, but all you need to know is that if you measure capacitance, you can use that value to calculate thickness. So our results, what we found was that if we incorporate caffeine into our system, we get a thickened membrane. In other words, we found that if we incorporate caffeine, the capacitance of the membrane is lowered when compared to the control. And that once again suggests that we have a thickened membrane. Now I just want to take the time to discuss the many experiences I've had as an undergraduate researcher at Iona and as a member of Project Symphony. In the top left corner, you can see all of the current members of Project Symphony, and this was at our most recent trip to an ACS national meeting. Over my past 4 years, I've been to 3 ACS conferences and to 1 national conference of undergraduate research, and we were actually scheduled to go again this April, but unfortunately we couldn't make it. And another experience I've had is I had an NSF-funded research internship with the University of Tokyo. I spent 5 weeks there over one summer, and I worked collaboratively with the group there to design new devices for our capacitance studies. In the bottom left-hand corner, you can see me with all of the group members at the lab in Japan. In the center, you can see me with the professor I worked one-on-one with, Professor Toshinisi Osaki. And in the top right-hand corner, you can see me with 3 other members of the lab where we took a trip to one of the islands of Tokyo. And this group not only just stimulated my scientific inquiry and gave me a better understanding of how global science is and how many research initiatives can be brought together to come to further advances, but it allowed me to understand other cultures, and it allowed me the ability to dive into a new culture and a new surrounding and learn how to adapt to it. Lastly, in the bottom right-hand corner, this was me at a conference known as Posters on the Hill, where I was selected to be a representative in New York State and advocate for undergraduate research. Over my past 4 years, I realized how important research has been in my life and my success as a student, and I wanted to make sure that all other students at other universities have the similar opportunities. So at this conference, I met with many legislators and government officials to convince them of the importance of undergraduate research and how the funding should be kept going that way. So in conclusion, I just want to say thank you to Dr. Lee. I want to say thank you to all the members of Project Symphony. Without you guys, I don't think I'd be where I am today. And I want to thank you for listening.